to visualize these types of fragments on the gel. Okay, and what you actually need to run on the gel as well is a standard, so you can actually determine the molecular weight. So you can go from millions of base pairs in your sample to down to thousands, so that's one way. Or you can even get single base resolution, where you're cutting one base at a time, and then you can separate all the bases. So this is what your gel would typically look like. And again, this is a nightmare to read when you actually want to determine the sequence. There's so many bands being separated on this gel. And again, the smaller fragments would migrate towards the anode because they're negatively charged. And again, we're going towards the positively charged region. Okay, so that's great. We can separate these fragments and we can look at the patterns and maybe identify some types of sequences. What is it good for? Well, it can actually help you if you have different samples of DNA to look at the similarities. Are there similar sequences within these different samples? So what you could actually do in the lab is take your sample and put it into 4 different test tubes, and then use 4 different restriction nucleases in each of those test tubes. Okay, so we have the cleavage patterns depicted here, what they will recognize, and then run those samples on a gel, and then compare it, same type of treatment, to a second sample. And again, the thing that would be able to determine from this is actually if there's any similarities between these cleavage sites from the one sample and sample number 2. So we would actually look to see if there are similar fragments being generated. I don't typically see anything that's very similar. So they're very different, distinct DNA samples that are being created, or fragments being created here. In the middle, you'd actually run a standard. So MW means molecular weight. And then the C over here is your control. So that's your undigested sample. So you would compare it to sample number 1 and then sample number 2. So these samples are very different, and they can tell you that you probably don't have very similar sequences that contain these types of restriction sites within your samples. So that's pretty good. So you can use this technique to do that. And then you could actually even take it further. You can use it in disease. So you can use that type of disease testing for disease diagnostics. And early on, before the advent of DNA sequencing, this was a technique that was actually employed for the diagnosis of cystic fibrosis, because it facilitated the examination of a mutation in the CFTR gene. So there's lots of mutations associated with cystic fibrosis, but one key one that came out was the R533. So this means the amino acid arginine in position 533 in the protein. Its codon was actually changed to a stop in the DNA. Okay? So there was a base mutation that resulted in that amino acid codon being changed to a stop. And ironically, or serendipitously, this mutation actually led to the disappearance of a restriction site. Okay? Sometimes if, again, you have a mutation, maybe a restriction site appears, or maybe it disappears. And you can use it as a diagnostic tool to identify carriers of this mutation. And I'll show you how that was actually done. So typically, there is a HEN2A restriction site in the CFTR gene, and I've shown it here. So here's the palindromic sequence. And that's present when we have the amino acid arginine in position 533. So if you were to take someone who was homozygous for just the wild type, so no mutations associated in the CFTR gene, and cleave it with HEN2A, basically you would get a cut and you would get a digested fragment, which is represented on this gel. Okay? When this amino acid, then its codon is converted to a stop, this site actually disappears. Okay? So if you were to treat somebody, say, who was heterozygous for the mutation, so they had a normal wild-type copy and then, again, a copy of the mutation where the site has disappeared, you would actually get 2 bands. One where the wild-type is being digested, so it's represented here, but for the mutated copy, there would be no digestion. So the band would appear here, very large. what would you see for somebody who was homozygous for the mutation? So if we had 553X and 553X, and we treated it with HIN2A, what would you expect to see on this gel? How many say all undigested? Yeah, so you probably just see something over here. So this is how they initially diagnosed this mutation in CFTR, in the CFTR gene. Okay? So you can apply it clinically and figure out if somebody is a carrier or actually expressing the disease. Okay, so that's great. We could do some diagnostics. We can cut up our DNA. How about synthesizing DNA? So around the 1960s, this is where they first started. Can we synthesize DNA? So this is where the question arose. So I'm basically going to go over the procedure of how we replicate DNA, which Dr. Brown will get into more detail. So hopefully you have some idea. We have our double-stranded DNA. Typically, you will need to denature the double-stranded DNA so that you free up a template for the DNA polymerase. We're going to add in some primers, and we need primers for DNA replication because it cannot start without a free 3' hydroxyl. Okay, so our primers will bind to the template and again also to the non-template strands. We can add in the polymerase and our nucleotides. We can radial label them, and then the polymerase will synthesize a daughter strand. And again, remember, DNA replication occurs in the 5' to the 3' direction, always, on both strands. So here we see our strand being synthesized 5' to 3', 5' to 3'. Okay, so these are our new strands with our primers. So this is what happens in nature when we synthesize DNA or we replicate it. In the 1980s, they actually developed a machine that could actually synthesize DNA, an automated system. So you put in your nucleotide bottles here, your polymerase, and again, your primers in your DNA, and you could walk away and synthesize some DNA. However, this procedure didn't use nature's methodology. So we're going to go through how they actually were able to synthesize it. Whoops. Again, here's just a representation of nature's reagents. We have one strand that we need to replicate. We have our primer that has that free 3' hydroxyl that's needed for replication. We add in our polymerase and our DNTPs. So here it's DATP plus our polymerase. And we incorporate a nucleotide and we release PPI. That's pyrophosphate, okay? And that pyrophosphate will actually then be hydrolyzed to 2-PI, which drives the reaction forward. Okay, so we incorporate the base. Again, we get another base incorporation, release of PPI, and we elongate the daughter strand when we're replicating the DNA. Okay, so that's nature. So basically, we're going 5' to 3', and we use D and TPs to do so. Synthetic DNA, or the production of it, is in the 3' to 5' direction. So we're going to go in 3' to 5' direction. And we're not going to use D and TPs. We're actually going to use modified nucleosides. And it's this one here. It's called a deoxyribonucleoside 3' phosphoamidite with DMT and beta CE attached. You don't have to memorize the structure, okay? So that's a good thing. But we're going to look at it just carefully. Instead of adding these DNTPs, we're going to add these types of deoxynucleosides to the reaction. So not only are we synthesizing the opposite direction, we're going to add these modified nucleosides. So the first modification is the addition of this dimethyl tritol group, so this DMT, which actually protects that 5' end, so that 5' hydroxyl, so the 5' end. so that it doesn't react with another 5' hydroxyl. So you protect that. You protect the bases as well, so you don't get any nonspecific interactions happening. You're going to protect the carbonyl on the phosphate again. You're going to use beta-cyanoethanol group, so this blue portion here. And then you're going to add on a phosphoramidite component. And this is the actual component that's used for the reaction to get nucleotide incorporation. Okay? After you've got nucleotide incorporation, we're going to remove that DMT to make a free 5' hydroxyl, which is needed for the incoming modified deoxyribonucleoside. And then at the end, you're going to cleave off the beta CE group and anything that's protected on the base when you synthesize the DNA. Okay, are we clear on this? We're going in the 3' to 5' direction, and we're going to use these modified deoxyribonucleosides to make synthetic DNA. Okay, so how does the reaction happen? Basically, we're going to get a solid support, so a resin, and we're going to attach our nucleoside. That's the last one in the sequence, right? So this is the 3' end. So this base represents the last base in your DNA sequence. We have a free 5' hydroxyl on this base, on this nucleoside, okay? All right, so it's attached to the resin, and we're going to synthesize everything in this direction. All right, so we have it attached to the resin. We got our last nucleic acid in the sequence attached to the resin, and then we're going to slowly start adding these modified or activated monomers onto that 5' end. So this is how we're synthesizing. 3' from this end to 5'. So we bring in our protected deoxynucleoside. A phosphoramidite component is depicted here in this NR2. So this is our phosphoramidite component. And this is where we have nucleophilic attack occurring. This is the reactive portion. It's a great leaving group, so it's going to leave after the reaction happens. So it comes off. And we get the formation of the phosphoester link that occurs between these two. Okay? Here's our BCE, that's protecting the carbonyl from the phosphate, and our DMT, that's protecting that 5' hydroxyl end. All right, so we get the reaction happening coupling, the NR2 leaves, and we get the formation of the ester bond. Then we will oxidize that carbonyl to create the carbonyl, the oxygen, through the use of iodine. Okay, so then we can create that carbonyl group, again, through oxidation, and then we deprotect that base, so the DMT comes off, leaving a free 5' hydroxyl. Then we repeat the cycle. We bring in our next modified deoxyribonucleoside and carry out the same procedure. Once we're done elongating the chain, and we've gone from the 3' to 5' direction, we cleave off all the BCEs and any base protection groups that we might have. And we basically synthesized our new DNA strand. Okay? Is everybody clear in how we do this? Two key differences. We go from 3' to 5', and we don't use DNTPs, but rather this activated monomer. And this phosphoramidite component is the actual leaving group that carries out the reaction in formation of that phosphoester link. Okay, so that's great. What can we do with that when we're synthesizing DNA strands? Well, we can actually use this type of procedure to create smaller DNA strands that we can classify as probes. So maybe you've heard the terminology. Probes are actually used in the lab when you carry out southern blotting And southern blotting typically involves, again, same type of thing. We would take our DNA and then cleave it with some restriction nucleases and run it on a gel so we get those different fragments. But rather than staining the gel with intercalators, which can be very toxic, you can actually transfer those fragments onto a nitrocellulose membrane. And you can do this in an alkaline solution, and just by straight through capillary action, those DNA fragments move from the gel into that nitrocellulose. So you have your nitrocellulose. You can't visualize anything on the nitrocellulose component here, but if you create probes, which can be very complementary to a specific sequence you're looking for in a particular fragment, you can hybridize these probes to that DNA fragment, and again, using radial label, you can actually detect the signal through an audio radiogram. So you can design these probes, again, being single-stranded, attach a radial label to it, so maybe P32, that could emit something that can be picked on a film, so you can get an audio radiogram, mix it overnight with your nitrocellulose membranes. If there's complementarity to a specific sequence in your DNA fragment, then you would get base pairing occurring, and you would be able to pick up that signal onto the film that you've incubated your nitrocellulose with. A good probe typically forms these double-stranded helices. Sometimes, if you design the sequence incorrectly, you can get a bad probe, which will, again, have imperfect base pairing. You still may pick up a signal, but it might not be as strong as something that has total complementarity to that that DNA sequence in a particular fragment. Okay, so you can synthesize these types of probes, short stretches of DNA that you can radial label, and then take the step further. Instead of using a gel, transfer your fragments onto a nitrocellulose membrane, and then detect complementarity using audio radiograms. Okay, so you can use this synthetic DNA process to generate these types of probes. And that's really great because you can take it a step further and use this technique to identify fragments amongst different DNA samples. And again, same type of procedure. Say we had a crime scene, so you can use it in forensics, and we had some blood that was left behind, and we can call it the evidence, okay? You have a victim and two possible suspects. So if you take the DNA from all four of these, and again, cleave them with restriction nucleases, you get your DNA fragments, you can separate them on a gel, transfer it to nitrocellulose. You can create your probes based on the sequences found in this DNA blood in the evidence, and then hybridize them to the nitrocellulose to see if you can pick up any similarities. And then you can get this blot right here through X-ray exposure to a film, and just by looking at the film, whose blood was it as part of the evidence at the crime scene? Was it the victim's, suspect 1, or suspect number 2? Compared to the evidence. Who's guilty here? How many say number 1? Yeah, he's guilty. Well, not he, sorry, she. Can be anybody. So they're guilty. Okay? So again, you can see the banding pattern is very similar to the blood that you see in the evidence, that you would actually see with the evidence undergoing these techniques. Okay, so you can use it diagnostically in forensics to carry out these types of procedures. Likewise, you can change it around a bit, and instead of using radioactivity, you can actually tag fluorescent modifications to your probes. So again, you can use these to identify presence or absence of genes in the genome. And it's really great because these types of fluorescent probes you can actually introduce into the cell, and you can look for hybridization to actual chromosomes. So here's just a representation of that using a fluorescence microscope. This has been used, this technique, for the identification of multiple copies of the HER2 gene. So here's just an example that's located on chromosome 17. These multiple copies, if you're greater, I think greater than 17 copies, of this HER2 gene, you have a higher risk of breast cancer. So that's one way to actually identify who's susceptible to breast cancer through this HER2 gene, if you have greater than 17 copies on this chromosome. Okay, so we talked about restriction analysis, how we can synthesize DNA, blotting, and hybridization. Well, what about DNA sequencing? Well, DNA sequencing back in the early days was really crude. I'll just introduce some of the methodologies. Again, they're not typically used today for a variety of reasons, which I'll explain. But typically, you would just take your DNA sample, radio-label it, so add on a p32 at the 5' end, and then subject that DNA to different various chemical treatments so that it could actually get cleavage of specific bases depending upon the treatment. So in this case, Maxim and Gilbert actually devised 4 different types of treatments that they could subject their DNA to, which would result in preferentially cleaving the G's off the DNA strand, a treatment that was specific, more specific for A's, another treatment just for C's, and then one that would just cleave both the C's and the T's in the strand. And these treatments typically involve using the compound dimethyl sulfate. Under neutral conditions, it preferentially reacted and cleaved the G's off. Dimethyl sulfate in acidic conditions would cleave the A's. Hydrazine with some sodium chloride is very specific for C's, so we get cleavage. And then just hydrazine alone, it would actually cleave both pyrimidine nucleotides. And then once you've done that reaction, to each test tube, you would add pepyridine. And pepyridine is essential because you cleave the phosphate backbone exactly where that specific base has been removed. So when you cleave that phosphate backbone, you can get different size fragments, and then you can run them on the gel. And because your DNA sample is radio-labeled, again, you can detect them through radiogram. So this is a very crude method. And initially, when they started sequencing DNA. And again, here we have a DNA strand, just a representation. If we have hydrazine, we place it in a solution of hydrazine, and we would cleave the pyrimidines, so anything in blue, the C's and the T's. If we added in some sodium chloride, it would be very specific and cleave those C's off. Again, you need the pepyridine to cleave the phosphate backbone where that base has been removed, to get a different size fragment. The dimethyl sulfate cleavages was, again, depending on the conditions, acidic or neutral, specific for the purines, so the thymidine and the adenine. So in this example that I have here, we have a strand, and typically, again, you would radio label, so you'd have your p32, that's what this black dot represents. This sample underwent treatment, and we can see that it's just specifically cleaving at the C's. Okay, so again, in the sequence, we're getting a cut there. And we get 4 different types of fragments, and typically that would be run in this gel. So then you can see you get 4 different types of banding patterns based on the size, the larger fragment at the top, and then the smaller one down here at the bottom. Okay, so this would be hydrazine with some sodium chloride. We take that sample and we incubate it with hydrazine and sodium chloride. We get 4 different fragments, run it on a gel. Likewise, you would take that sample and do your other conditions and do the 4 comparisons on the gel. Again, how do you read this? How do you read the sequence now? Basically, sequences were read from the bottom upwards, and this would be the 5' to 3' direction. And the problem with this, remember, hydrazine and dimethyl sulfate, sometimes it would cleave specifically for one nucleotide or non-specifically for both. So how do you read this? So you would start with the furthest migrating band. So we can look in this column. We can see the treatment was non-specific for either A's or G's. But if we compare it to the G column, we don't see a G. So basically, this band will represent A. And we can name it an A, an adenosine nucleotide. So that's the first base in the sequence, which we see up there. Then we'd go to the next band that we see as we're moving upwards. And we see two bands that are at the same level of migration, one in the T plus C and one in the C. So because we have a C here in this position, this is probably just representation of C. So that would be the next nucleotide in the sequence. So again, if we look up here, here's our C. Then we move over here, and we see again two bands, comparisons between the G plus the G plus A. We see a G, so this band more than likely represents a G nucleotide, and we can put that in the sequence. Finally, if we get to this one up here, in the T plus C column, we see a band, but we don't see anything in C, so this means this must be a T. And then we can read the entire sequence going from the 5', 3' direction, and hopefully it gives you the sequence that you actually see up here. Okay? So this procedure is typically not done anymore in the lab. I mean, this compound is very, very toxic. So it's highly combustible. It's known to be carcinogenic, so I definitely wouldn't want to be working with it in the lab. But again, just to show you what types of techniques they were working with in the early days to sequence DNA. Okay, so Sanger then came along and said, well, you know, that's an interesting way to sequence DNA, but I'm going to take advantage of the actual synthesis of DNA and develop a methodology that can enable DNA sequencing. And he developed this methodology in 1977, and it's called chain terminator sequencing. Okay? So again, it takes advantage of your DNA polymerase reaction, in which we got our template strand, we got our primer, we got our DNA polymerase, we got our dNTPs. We can make DNA. But at low concentrations, we're also going to add in dideoxynucleotides to stop the reaction. So here's our deoxy-NTPs, so what we normally would add into the reaction to carry out DNA synthesis, or dNTPs. Here's the dideoxynucleotide. What's the difference between the two? We're lacking that 3' hydroxyl. So dideoxy, so 2 times, we're removing that hydroxyl group. What's the problem with this if this base gets incorporated during DNA synthesis? Why does it stop? Why does it terminate synthesis? What did I tell you about DNA synthesis? What do we need? A free 3' hydroxyl, which is lacking here. Okay, so you don't have that 3' hydroxyl for synthesis to still occur as you're synthesizing DNA. Okay, so based on this, he developed a strategy where you would actually take your strand that you want to replicate. You would add in your primer. Again, this is radial labeled. You would set up 4 different test tubes. You would add in your dNTPs, and these would all be in excess. So all of your dNTPs, so the regular ones. And then with each of those 4 test tubes, you add in the dideoxynucleotide. So this one would be ATP, ATP. This one in this tube would be for thymine, cytosine, and guanine. And again, these are at low concentrations. Okay? So these are at low concentrations. Whereas your regular dNTPs are in excess. So then you add in your polymerase, you got your primer, you got your template, replication starts happening. Because these are in excess, you may start synthesizing a very long chain, and then all of a sudden, one of these dideoxynucleotides gets incorporated, synthesis stops. Polymerase starts again. You get incorporation of your regular nucleotides, and again, maybe a shorter fragment occurs because the dideoxynucleotide gets incorporated sooner. Maybe right off the bat, the polymerase incorporates the dideoxynucleotide. So you get these shorter, shorter, and shorter fragments. And then you can run each of those fragments that are generated on a gel. Again, it's very specific because you've labeled which ones, where you have those dideoxynucleotides. And again, you can read the sequence going 5' to 3'. Right? And note that this sequence that you're reading is complementary to your template, right? So it's opposite. It's a key thing. So how would you read this? What's the sequence of this? Just start off. What's the first letter? A, T, G, T, C, and so on. Okay? So you read it that way. So if I gave you one of these gels on the exam, asked you what the sequence is, you should be able to read it for me. Okay, so that's great. Again, we want things to be automated. We can put it into like a specific machine, walk away, go have our coffee. So they started developing equipment that could actually do this automatically on its own. Again, the major difference here between Sanger sequencing and this automated DNA sequencing was that rather than using a gel to read off those fragments, they actually developed a machine in which those fragments were actually passed through capillaries or thin fibers. And it's called capillary electrophoresis as opposed to gel electrophoresis. And rather than using the radiolabel for your newly synthesized DNA, the base terminators had fluorescent tags attached to them. So you would add a fluorescent tag, depending on the color that you want, to the dideoxynucleotide. So as these fragments are passing through the capillaries, or these thin fibers, the machine would be set up to shoot a laser through these thin fibers. And then if a base terminator with a specific fluorescent tag was incorporated, you would get a color change, which could be detected on the camera in this machine. And then it would be recorded as this peak on the screen, so you could actually see it. And then depending on the color that you have and the actual height of that peak, you could determine the sequence or read off the sequence. Okay? You don't have to worry about the actual mechanistics of how this machine actually works, but know rather than using gels, you can use capillaries and actually fluorescently label your base terminators to be able to get one of these diagrams on a screen to be able to read the sequence. Okay, so in the early days, again, what they started working out with were very small genomes, so there weren't a lot of base pairing numbers associated with them. It took them many years to be able to sequence these genomes from these species here. It was only by the late 2000s that we finally was able to sequence the human genome, so in 2001. So it took almost a decade from when they, almost 2 decades from when they first started out to able to sequence something that has, again, more than 3 billion base pairs, so you can see the size. And again, look at the difference in terms of how many years it actually took, going from something very small to something very large. And we'll look at the techniques that actually enabled them to determine this type of genome sequencing. And then, I know you probably see this on the news, you know, these DNA kits you can buy, sometimes they're called, you know, from 23andMe. You can actually obtain them, send in your DNA sample, and maybe determine some type of ancestry or maybe some prone to some specific disease. These DNA kits, they're not really sequencing your genome, they're only doing parts of it so that they can figure out if you have any known diagnostic inheritance patterns or susceptible to any specific diseases. So don't be fooled into thinking they're actually sequencing your entire genome when you send out for these kits. Over the years, the DNA sequencing efficiency has increased dramatically, and it basically follows Moore's law, which states that computer efficiency tends to double every 2 years, and because it doubles every 2 years, the cost associated with it also comes down. So you can see when they started off with those smaller genomes, it took approximately 13 years to sequence them, and look how much money it actually cost. Okay? Now, I guess in the last decade, we've gotten down to about 2 weeks, so sequence the entire genome, and it's almost less than $1,000 to do it. Okay, so it basically follows that principle. And we'll look at how these techniques developed over the years. Okay, some things to consider when you're in the lab and you want to sequence a particular DNA sample. Again, cost per megabase. So you can see how the cost again, has come down over the years, depending on the new techniques. And we're going to look at each of these individual techniques separately. So we've got single molecule sequencing, protein flow, and pure sequencing. Time. Time is of the essence. You don't have 13 years to do your PhD to find out a particular DNA sequence. So some of these methodologies, if you look at Sanger sequencing, took 2 hours. Some take longer now. Reading the length. Again, size was a big factor in being able to sequence DNA. Now we can work with up to 1 million base pairs, and especially with the pure sequencing technique. And the cost of instrumentation, again, is another factor of when you want to sequence your DNA, what you're going to use. Okay, so the key thing about these methodologies, so we're going to go through the 3 of them, the single molecule, the proton, and the pure sequencing. Don't worry about the complexities of how these readouts are obtained, but rather the principle behind them. Okay, so again, we can think of DNA replication. We have our polymerase, we've got our template strand, we've got our primer. In this type of sequencing, your nucleotides have a fluorescent tag attached to them. So if we incorporate the correct nucleotide, we get a burst or a pulse of that fluorescence, which is then detected on the machine, on the screen, and it's emitted as this intensity reading, okay? And depending on what color you attach to which nucleotide, you will be able to determine what nucleotide was incorporated. And again, if there's a nucleotide incorporated, that fluorescence gets cleaved off. We then flow in more bases, and then if another incorporation occurs, if we have an A with a blue tag, we would get another pulse. Key thing about this procedure is you flow in each individual nucleotide one at a time. Okay, so you'd flow in some C's. If you get incorporation, that's great. If you don't, you would wash them away and then flow in some A's and see if you get a pulse reading. Okay, so that's single molecule sequencing, basically adding a fluorescent tag to your nucleotide. Once incorporation happens, that gets cleaved off and the signal is detected. Proton sequencing. Now this takes advantage of the actual reaction. And if you remember when I told you, if we incorporate a nucleotide into our growing nucleotide strand, we release PPI, so this is pyrophosphate, so PPI, but we also release a proton in the reaction. This methodology takes advantage of that proton release, because these protons can actually generate electric current, which can be converted to a voltage, and we can use an ion sensor to actually measure the amount of protons that are being released. Okay, so basically just another representation of that. If we have our nucleotide come in, if it's not complementary, no protons released. If it gets incorporated, we release a proton. And again, if we have the same one that comes in that gets incorporated, we can release 2 protons. So that means 2 nucleotides, the same base, have been incorporated, and we get twice the amount of protons being released. Same principle as single molecule. You flow in the nucleotides one at a time. If you get incorporation, that's great. If you don't, you wash them away, and then you flow in the next incoming nucleotide. Okay? So if you get multiple bases being incorporated in a row, you release multiple protons. These protons, the current associated with them, can then be detected by the machine, and you can get these peak graphs being observed on the computer. So just for example, say the blue was a G base that we flowed through. We didn't get incorporation, you would not pick up an electric current signal. Again, if you flowed through an A, you probably wouldn't get anything. If you flowed in some C's, maybe you get twice the amount of current being associated with this incorporation, and you can say that 2 C's were incorporated into the sequence. And then finally, if you flowed in a T and you only got half the amount of current, you can say that only 1 T was incorporated into the sequence. And then you can read them across off of the screen, depending on that current that's being observed or recorded. And finally, this pure sequencing. Again, this takes advantage of the actual incorporation of the nucleotide. And rather than using the proton that's released, it actually uses the PPI. So the PPI, in the presence of sulfurase and APS, which is adenosine phosphorosulfate, you don't have to know the name, can combine to form ATP. Okay, so if a nucleotide gets incorporated, we release PPI, and we can form ATP in the presence of sulfurase. This ATP concentration is proportional to the amount of nucleotide incorporated. ATP can then be used by luciferase in the presence of luciferin to create oxyluciferin, which emits light. And again, this is proportional to the amount of ATP synthesized. And then you can use a detection program to actually detect the amount of light that's being released. So you can put your DNA sample in a well, add in the polymerase, your DNTPs. Again, you're going to wash them through one at a time, your sulfurase, your luciferase. And if you get base incorporation, you're going to have this reaction happening and the well emitting a light, which can then be recorded on a computer, so for detection. And again, same thing, you're washing the bases one at a time, okay? Taking advantage of the pyrophosphate that's being released, so you get pyrosequencing, and an indirect measure of the amount of ATP that's being synthesized. All right, so just to summarize, that's the next generation sequencing techniques. So you get high throughput sequencing based on the biochemical reactions. The direction is achieved using optical or electron readouts. They're a huge improvement over Sanger and Maximum and Gilbert, both in cost and time. And now we can sequence numerous sequences in large genomes in order to understand their biological function. All right, so we talked about DNA sequencing. In the last couple of minutes, we're going to talk about, hopefully get through PCR, and maybe we'll reserve this for next day. Okay, has anybody done PCR in the lab? Okay, so hopefully this is an easy slide. What do we need for PCR? Given what I've told you about for DNA replication, what do we need? Do we need a DNA polymerase? Yes. Typically, the DNA polymerase used in PCR is Taq polymerase. And this is actually was discovered in hot springs. And it's very temperature resistant. Okay, so that's important because of the nature of PCR reaction. Okay, what else do we need? Yeah, we're going to need primers. We're going to need our DNA strand. And what else? What do we need? DNTPs, right? What's the methodology for PCR? What do we do in PCR? What is it for? What are we doing with our DNA? Amplifying, right? So we're just going to do an amplification. So we're going to take our double-stranded DNA. So we need to separate them. So what are we going to do in the reaction? We're going to add heat. So we get two strands. And then we're going to let the DNA polymerase plus the primers and the DNTPs make copies. So basically amplify. And then we repeat times X number of cycles. Okay? And usually this amplification is exponential. Okay, so you're going to do it multiple rounds. You take your double-stranded DNA, separate them, your primers can anneal, you have the polymerase, your DNTPs, and you can carry out replication, make multiple copies. You take that double-stranded DNA again, you heat it, separate it, and go on and on. Okay, so you can do amplification. Does everybody have this? So what is PCR good for? It's really a good diagnostic tool. It can definitely amplify regions of DNA that may vary from one individual to another. So we have variable regions on our chromosomes, which come in useful for identification purposes. We can use it to clone pieces of DNA if we use our primers that are targeted to specific sequences. And we can introduce mutations, so we can do mutagenesis. And we'll look at an example. And we can develop real-time PCR so that you're actually measuring in real time the amount of DNA that's being synthesized, hopefully to detect any specific sequences that might be there. So let's just look at one diagnostic tool before we go. In our chromosomes, we have a lot of variable regions, and these variable regions are sometimes called short tandem repeats, which are depicted here in gray for 3 different chromosomes for 3 different samples, so individual A, B, C, and D. And again, they're also known as variable number of tandem repeats. They're basically just very short segments of tetrameric nucleotides that repeat over and over and over again. And depending on you as an individual, the lengths can vary. So you can design your primers that are specific to these sequences in these variable regions, and then amplify that particular product, run those samples on a gel from these 3 different chromosomes, and you get the different colors represent the 3 different variable regions, and compare them against other samples. Typically, if the sample is the same, you have the same type of fragments that are being generated. So from this, again, it can be used in forensics, this forensic sample F, which individual is responsible for that forensic sample, just by looking at it. Which one? B. Yeah, so B has the same type of banding pattern. So again, more than likely, this sample came from this specific individual. And what's interesting enough is that there's actually a database that's been established in the US, and it's been shared by Canada. It's called the CODIS database, or the Combined DNA Index System, in which the repeat motifs, 13 core of them, have been identified on these chromosomes. And you can actually take your DNA sample and compare it to, I guess, well-known crime scenes or known offenders and see if you can get a match. So you can actually go to this database if you have a forensic sample that you need analyzed. Okay, so we'll finish off the rest next day. Don't forget the quiz on the weekend, and we'll see you next Friday.